Good afternoon. My name is Gina Wen. I'm a SMID-cap biotech analyst from Barclays. It's my great pleasure to introduce our next speaker, Victor Perlroth, Chairman and the Chief Executive Officer, and also together with us during Q&A session, also Jason Ehrlich, Chief Medical Officer and the Chief Development Officer.
Great. Thank you, Gina, and Barclays for having us here at the conference. Thanks also to the webcast audience. I'm Victor Perlroth, the CEO of Kodiak Sciences. Our forward-looking statements. A quick look at our catalysts before we jump into the Kodiak story. So in the first half of 2019, we plan to complete enrollment in recruitment into our phase 1b multiple dose study in patients with wet AMD, diabetic macular edema, and retinal vein occlusion. We'll also start the global pivotal phase 2/3 study, which is a head-to-head of KSI-301 versus Eylea in treatment-naive wet AMD patients. In the second half of 2019, we'll plan to present ongoing phase 1b data at key ophthalmology meetings and investor conferences. We'll also initiate China studies in wet AMD and DME, disclose more around our earlier non-proliferative diabetic retinopathy strategy, and initiate global pivotal phase 2/3 study in DME. We're also planning for an R&D day, haven't scheduled that yet. First half of 2020, be looking towards continued phase 1b data, and in the second half of 2020, looking at interim data coming out of our global phase 2/3 study in wet AMD, as well as interim data from other phase 2 studies that will be ongoing. That's towards the end of 2020. Kodiak is an ophthalmology medicines development company with a singular focus in ophthalmology with a global scope, from discovery through commercialization. Our emphasis is on high science medicines design for products and platforms for high prevalence ophthalmology, and to apply that high science to develop what we call go-to medicines, or the agent of choice in each indication. And to not just build individual agents, but rather to build a pipeline with multiple shots on goal across the major high prevalence indications in ophthalmology, wet AMD, dry AMD, and glaucoma. We have strong leadership in the company, a management team with experience to build a leading biotech company from the operating standpoint. Also from the board of directors, deep biotech and governance experience. And from a scientific and clinical advisory standpoint, working with a number of recognized ophthalmology leaders, also on a global basis. So what's the problem to be solved? Well, intravitreal anti-VEGF agents and their anti-VEGF mechanism of action have been shown to improve and maintain vision in patients when dosed per label. So early visual acuity gains, as we see here on the bottom left, and those gains can be sustained over many years when dosed per label. For example, Lucentis on a monthly basis, or Eylea on an every other month basis. So the mechanism is solid, and so is the concept of anti-VEGF biologics as an intravitreal therapy. Yet, minimal visual acuity gains are achieved in real-world practice. So without continued high-intensity treatment, vision loss begins after only 3 months of anti-VEGF therapy. This is seen now in numerous studies and evaluations. It's seen in Europe across a range of different countries. It's also seen in the United States, and it's also seen with all different current agents. Why is this? Well, current anti-VEGF drugs do not control the disease for long enough between doses. As you see on the right here, we can see the traditional, with Eylea, sawtooth pattern of Eylea dosed on its every 8-week label, where we have recurrent swelling of the retina, which happens between 4 and 8 weeks on average in patients, demonstrating recurrent disease activity, which pops up in between doses. Overextension of a treatment interval or under-treating the patient that allows drug holidays in between injections leads to disease progression and permanent retinal damage. So what is needed is a new crop or a new agent that that's an anti-VEGF medicine that allows to control the disease for many months, i.e. long enough to sustain, to keep disease activity at bay between less frequent injections. Yeah. Yeah. That's what Kodiak has designed. Our antibody biopolymer conjugate, or ABC platform, built on antibodies as biologics with stable linkages to novel phosphorylcholine-based biopolymers to build ABC platform medicines. These medicines are the same where it matters, but different where they need to be different. By the same, they're targeting the proven VEGF epitope, the same as Lucentis. They're antibody-based biologics. They're intravitreally injected, and they're formulated as optically clear aqueous solutions. They're injected in the same way. They traverse the eye and the ocular tissues and access the retina to have their action, and then they leave the eye in a similar manner as the current Lucentis or Eylea agents, with no ocular residues. Yet they're different where it matters, bringing the high science to deliver ocular durability that's in a class of its own, with fast systemic clearance, improved bioavailability to the retina and choroid, the tissues of action, and improved biocompatibility and stability. KSI-301 is being designed and developed to be the go-to anti-VEGF agent to solve this real-world problem for patients on a global basis. Next slide. Here we see the science around the design of Kodiak's lead candidate, KSI-301. Its anti-VEGF conjugate versus the predicate agents ranibizumab and aflibercept. We see the size of KSI-301 is 950,000 molecular weight versus 50,000 for ranibizumab or 115,000 for aflibercept. The larger the agent, the longer the dwell time in the eye. At the same time, we formulated KSI-301 to a high formulation strength, and our clinical dose that we've chosen based on our phase 1A study is 5 milligrams. At that formulation strength and with that design, we're dosing 7x the moles of ranibizumab, and we're achieving an ocular half-life 4 times longer, which translates into 1 million times higher concentration of KSI-301 at the 3-month interval versus, say, Lucentis. With such a large molecule, 950,000, and with an antibody together with this stably linked biopolymer, some questions emerge: would it be potent? Well, actually, KSI-301 bioconjugate has shown greater potency in vitro than unconjugated anti-VEGFs. And in fact, it's more potent than the unconjugated starting protein. Furthermore, with such a large medicine, is it actually going to penetrate and get to the retina and the choroid, the tissues of action? We've shown that KSI-301 bioconjugate, because of the special nature of its phosphorylcholine biopolymer and the bioconjugate design, actually drives an enhanced bioavailability into the retina and the choroid. So despite being 8-fold larger than aflibercept, we actually have an 8-fold greater access to the retina. Further, because of its large size, KSI-301 bioconjugate has a flatter or better ocular PK curve versus the predicate agents, Lucentis in orange and Eylea with the squares there. We're in a class of our own as we think about ocular PK and durability. This translates into a large concentration advantage of KSI-301 versus other biologics over time. When we look at a matrix in comparison of our agent and technology versus current agents and other agents in development, KSI-301's high science design positions us very well versus competing efforts. Again, KSI-301 is the same where it matters and different where it needs to be different. The same, meaning we're an intravitreally injected biologic in an aqueous formulation at high formulation strength, and different where it matters in terms of our potency, tissue access, and anticipated durability improvement. As an intravitreally injected biologic as a next generation therapy, we're in a class where over 26 million intravitreal injections were performed globally in 2018. We believe, based on our design and our early data and the performance and trajectory and the clinical development plan that we have, that KSI-301 can be the single go-to anti-VEGF for all patients with retinal vascular diseases, encompassing all stages of therapy, as its use as an induction therapy, or its use as a maintenance therapy, its use as a supplemental therapy or a foundation therapy. In the context of being the go-to anti-VEGF agent, we believe KSI-301 can meet the individual needs of all stakeholders in the existing value chain, and we look forward to progressing this analysis together with the investor community over the course of the next year. We'll take a quick look at the phase 1 clinical data. Our phase 1 study, which data we announced in December of last year, was a single ascending dose study in DME patients, 3 different dose levels in 9 subjects conducted in the United States. It was a single dose with observations to 12 weeks and no retreatment. Patient information and demographics, it was a salvage phase 1 type population with very sick patients, as is usual in early phase 1 studies. Safety outcomes, every dose level was well tolerated through the 12-week follow-up period. There were no dose-limiting toxicities, no drug-related adverse events or serious adverse events, no intraocular inflammation of any form, and it was optically clear media after each injection. There were no anti-drug antibodies detected in any patient, and systemic levels of KSI-301 were surprisingly low. Improvements in vision and retinal thickness after the single dose of KSI-301 were seen through the 12-week final visit in the study. What we see here is vision in blue, higher is better through 90 days, and on the bottom, in dark blue, the OCT or thickness of the retina over 90 days. Lower is better. What we see here is rapid high magnitude responses as early as 1 week after dosing, with durable improvements out to 12 weeks. This is median change pooled across the 3 dose groups. What we really see here is a single injection of KSI-301 in a difficult to treat population. DME has a very high VEGF drive and is really a global disease in the eye. Here, with a single injection, what we see is a profile that looks like 3 to 4 injections or loading doses of an Eylea or a Lucentis. But we see that here with a single injection. We'll look at 2 quick case examples. In this patient, we saw, in this patient with severe swelling at baseline, who had actually failed through several recent injections of bevacizumab with worsening vision, had complete resolution and sustained resolution of his macular edema through 12 weeks, and flatlined on the OCT and 20 to 30 letter gain on vision based on a single injection of KSI-301. In a very different kind of patient, this patient with, looking here at the fundus photograph on the right, with very severe disease and extensive foveal lipid exudates, and also again a patient who had failed through bevacizumab, what we saw here was an early improvement in vision, about 5 letters, that was sustained through 90 days. But interestingly, we see on the OCT that it took about 30 days, and then from 30 days through 60, there's improvement on the OCT, and then further improvement from 60 days to 90 days. That's a very unusual pattern. That's not really seen with the existing anti-VEGF agents, and is suggestive that perhaps KSI-301's ability to penetrate the retina and the choroid and to really stay there and to drive improvements in the VEGF mechanism of action in a different way for patients. So we look forward to learning more about some of the unique features of KSI-301 and its science in patients. Having reached today, we've shown, and many important early development questions have been successfully addressed. We're now are active in our phase 1b open label study. It's now recruiting. It's going to explore KSI-301 safety, bioactivity, and durability in more than 50 patients in anti-VEGF treatment-naive patients as opposed to salvage patients. We're looking at 3 different indications: wet AMD, DME, and macular edema following retinal vein occlusion. All patients will receive 3 loading doses, followed by indication-specific reevaluation and retreatment criteria. So we're very excited to be recruiting this study. We look towards completing the recruitment in the first half of this year, and then presenting on an ongoing basis the data from this study, starting perhaps at ASRS in the summer, and then moving towards American Academy of Ophthalmology in October. We're also rapidly moving to be active in our phase 2 randomized controlled study of KSI-301 in treatment-naive wet AMD in the US and Europe. It's a pivotal study design, head-to-head against standard of care of aflibercept. All patients will be on Q12 week or better therapy, as infrequent as Q16 or Q20 weeks dosing with KSI-301. versus Eylea non-inferiority as a pivotal study. We're on track to begin enrolling in the second quarter of this year with an interim readout in 2020, looking at our durability proportions, and we can talk more about that in the breakout, and looking at primary readout in 2021. So key takeaways: KSI-301 is a novel antibody biopolymer conjugate that inhibits VEGF. It's been designed to be the same where it matters and to be different where it needs to be different to really solve the problem. Our phase 1A single ascending dose study results showed that KSI-301 was well tolerated at all dose levels. We've selected our top dose to move forward into pivotal studies. What we saw was rapid onset, high magnitude improvement sustained to 12 weeks in a difficult to treat DME population. Kodiak is executing on a comprehensive and aggressive clinical strategy for KSI-301 in retinal vascular diseases, broadly speaking, with phase 1B data to be presented at numerous scientific and investor conferences through 2019, and initiating multiple phase 2 pivotal studies in 2019 as well. Our objective as a company is to develop go-to anti-VEGF agent with KSI-301, encompassing all treatment needs for VEGF-mediated retinal vascular diseases, while at the same time building a broader ophthalmology medicines development company with a number of pipeline programs targeting other high-prevalence diseases in retina, such as dry AMD and even some of the retinal needs of glaucoma. Thank you very much.
Thank you, Victor. That's a very helpful overview. So Victor, Jason, so maybe I will start with a question on the PK measurement. Wondering, did you have any PK data in human to validate, the improved stability of 301?
Right. Well, certainly durability is a critical parameter of what we're building and designing towards. In our phase 1A study, we have measured systemic exposure data, but I think in the context of the large biologics such as Avastin, Eylea, and KSI-301, the systemic exposure data is less important from a PK standpoint because it reflects exit from the eye as well as your systemic compartment. So it doesn't give you very much visibility. I think what's important in our phase 1B study, we are looking at aqueous humor taps in patients, and that will begin to give us actual ocular PK data. Generally, that's highly variable with a lot of population variability in patients. We think that looking at the OCT, mean or median OCT, over time after injection will be an important way to look at durability, as well as like we saw in our phase 1A study, right, looking at the OCT over time will be important. So, we do think that the ocular PK is going to be important. We are generating that data. We expect that it could be highly variable when we begin to evaluate it, but we think it can be directional and important, and that on top of OCT data over time will give us a better view of durability. And then, of course, when we look at, say, our phase 2 pivotal and we look at the durability groups and the proportions, that's another way to gauge durability.
Mm-hmm. So regarding the aqueous humor biopsy, how frequent are you thinking to collect the samples?
I think we're doing it as frequently as it's a challenge, right? Because the physicians have a balance of wanting to generate the scientific data for curiosity, but not wanting to put the patients at risk. So we have a balanced approach.
Yeah, so we're in the phase 1B, for basically the way that it's set up, we try to get serial taps on patients until they need to be redosed, because we don't want to do unnecessary procedures. I mean, it's hard, as Victor said, to get patients to sign up for that. So, the idea is to try to generate as good a PK curve as we can from the aqueous humor, from collecting it serially on individual patients.
Right. And that's a little bit different. Hasn't been as often. That's new. Generally, people have looked more at a population basis, whereas we're trying to actually follow serial taps in patients, which has been done in maybe a few patients on Eylea. Actually, Diana Do, our chair of our scientific advisory board, was the lead author on some of the work with Eylea with Regeneron in serial taps.
Okay. And then you present the phase 1 data in the DME. So there appears to be a slight reduction in visual acuity, I think around 12 weeks if we compare to the 4 weeks. So, the benefit of the central retinal thickness was maintained. So just wondering, what could be the reason leading to that?
Yeah, so I think be careful not to overinterpret the vision data. So, visual acuity is very variable on a patient-by-patient basis. So, I'd say that like 3 letters from like one visit to another could easily be just noise, especially in a 9-patient group, right? If you have hundreds of patients, then, that's different. But in 9 patients, it's probably just patient-to-patient or visit-to-visit variability. You can take, if you or I went to the eye doctor and you looked at the eye chart, on one day versus another day, depending on like how dry your eye was and how much air conditioning was in the room, your vision could like fluctuate a lot. So.
Okay.
Yeah. So I think that to your point, the OCT curve that showed the stability in the median is maybe a better way to be looking at sort of anatomic durability, and vision ultimately follows that.
Okay. And then for the phase 1b data. How would you share with us later this year? In what kind of format, and then what time frame are you thinking about that will share additional data with us?
Right. Well, it's really 3 little studies at this point, I would say. So it's going to be north of 50 patients. So the RVO will be, say 10 patients, and the wet AMD will be around 20, and the DME will be around 20. And then based on the enrollment, and looking at different cutoff time points at, say, ASRS and AAO, that will help to provide some view of the type of data that we'll be presenting. So, it has 3 loading doses in all patients, and then basically it goes out 6 months. And so that will provide, I think, some very interesting data and a lot of interesting science. And we have some of the aqueous tap data as well as OCT angiography, as well as, of course, the vision as well. So I think we'll, be evaluating the data and trying to present it in a thoughtful manner through a series of, KOL meetings as well as investor press releases, and we're planning to do an R&D day in the second half of the year as well.
Okay. And then, for the phase 2 study in wet AMD, have you met with the FDA regarding, the trial design you just proposed? And also, what would be the non-inferiority margin you've been thinking?
Yeah, so Kodiak's discussed many of the aspects of the trial design with FDA. In terms of the non-inferiority margin, our understanding is that for comparisons against Eylea in AMD, it's a 4-letter margin. For comparisons against Lucentis, it's 4.5 letters, and that's what I think almost everybody is doing in studies that are running in the U.S. and Europe right now. So we're currently planning for a 4-letter non-inferiority margin against Eylea in the phase 2 study.
Okay. So you will start the trial in second quarter. What do you need to do now until you can start the trials? Yeah.
Yeah. So I think we're trying to design and implement the study as a pivotal study, as part of a pivotal package, right? So there's a lot of moving parts that all have to come together to do that, to execute at that level of rigor. So, the team is pulling out all the stops that can be pulled and is working really hard to try to make that happen. Obviously, there's a lot to do, there's a lot going on and to do.
So maybe going back to the clinical trial design, can you just, elaborate the purpose of the interim analysis?
Yeah, so the way that we're thinking about the interim is to look at, to have the data monitoring committee look at the durability proportions in the 301 group. So, for instance, the percent of people on 12, 16, or 20-week dosing at some given time point. And that, the amount of data will be determined based on enrollment into the study, right? So we're providing some flexibility as to when specifically we'll do it based on how well the study recruits. And I really think of it as it's an administrative interim, right? So it'll help us, ungate either additional investments in clinical studies or manufacturing for future clinical studies. There's a lot of things you can use with that information. But the main, I'd say a main component of it is to get actionable information, but not necessarily have to look at the primary endpoint, because we'd have to take a big alpha hit for that. And you can negotiate what level of alpha you would want to hit you'd want to take if you look at the primary with FDA, but we'd rather not have to look at the primary and potentially compromise the integrity of the study in that regard.
Right. So, I mean, our objective is to determine how many weeks worth, 28 weeks or 44 weeks worth of data on how many enrolled patients in KSI 301, right? And then to look at not the primary endpoint, because we don't want to take the alpha hit on vision, but rather look at the durability proportions. And we plan to do that both, hopefully, in our China studies that are, quote-unquote pivotals, as well as our global pivotal studies.
And then last question from me. So what will be your, thoughts on the trial design and the timeline for 301 in, DME and the other indications?
Right. Well, we're particularly interested, I would say, DME is a really important indication. We've already shown very nice data in it, even on a single dose. Of course, remember that EYLEA's label is 5 loading doses and then moving into less frequent dosing. So we think there is an interesting opportunity in DME to have a very similar study design as we might have in our treatment-naive wet AMD study, which would be industry leading. We're also, of course, interested to push beyond the DME, really even into sort of earlier non-proliferative DR, right, following from Panorama, but really following from a profile where one of our injections looks like multiple injections of, say, an EYLEA, and really opening up NPDR as a different type of indication that can really be dosed, can really build that market in a unique way and follow the market building of Roche, and Regeneron. So, and if you look at our phase 1b study, right, we have 3 different indications in there. We have the RVO, we have the DME, and the wet AMD. And so we're looking at KSI 301's clinical development build as really all retinal vascular kind of diseases, and thinking about, from a financial standpoint and an execution standpoint, and working backwards from when we might launch to figure out what our package needs to look like to have, all of that available. So, and I think we want to have a thoughtful, the go-to profile, right? As I said, we don't want to be a foundational therapy. We don't want to be just an induction or a maintenance. How do we develop it so that we can be all those indications and be, the leading agent for, because there's not going to be that many VEGF agents on the shelf in the future. We need to make sure that we're one of them. And that's how we're thinking about the development.
Thank you very much, Victor and Jason.
Thanks, Julie.
We also have John sitting in the audience. So we will have a breakout session across the hall.
Great. Thanks, Julie.
Great. Thank you.